For the longest time, the story of life seemed pretty simple. Our instruction manual was written in DNA. But what if the most important parts of that manual weren't in the main text at all, but were actually hidden in the margins? Today, we're diving into a hidden world that is completely rewriting everything we thought we knew about our own biology.
I'm sure you remember this from high school biology, right? The central dogma. It's this simple, straight line. DNA has the blueprints, RNA comes along, acts like a messenger, and makes a copy. Then that message is used to build proteins. The tiny machines that do all the work in our cells. For decades, we figured, hey, that's pretty much the whole story.
But what if that humble little messenger, RNA, wasn't just delivering messages? What if it was secretly running the entire show from behind the scenes? To really get this, we need to take a closer look at our own genome, specifically at these huge regions that, for a long time, scientists just kind of brushed aside as junk DNA. So, when we first sequenced the human genome, we were faced with this absolutely staggering mystery. It turns out only about 2% of our DNA actually contains the blueprints for proteins. The other 98%? Yeah, for years it was dismissed. But it turns out this isn't junk at all. It's the source code for a massive, hidden regulatory network. And the main thing that comes out of this so-called junk DNA is something called non-coding RNA. These are RNA molecules that never, ever get turned into proteins. A better way to think about them is as the cell's operating system, the complex software that's telling the hardware, the actual protein-coding genes, when to turn on, when to shut off, and exactly how to behave. All right, so who are the key players in this hidden network? Let's start with the unsung heroes, the real essential workers known as housekeeping RNAs. These are the guys that keep the cell's most basic processes up and running. I mean, even the most basic protein production completely depends on these non-coding RNAs. First, you've got transfer RNA, or tRNA. Just think of it like a fleet of little delivery trucks, bringing all the raw materials, the amino acids, to the construction site. And the factory itself? Well, that's built from ribosomal RNA, or rRNA. It literally forms the core of the ribosome, the machine that actually builds the proteins. Without these two, the whole system just grinds to a halt. But beyond these essential workers, there's this whole other class of non-coding RNAs that are way more subtle, way more sophisticated. They're the regulators. And let's start with the small but incredibly mighty microRNAs. So if turning a gene on is like flipping a light switch, then microRNAs are the dimmer. See, they don't just make simple on or off decisions. They give the cell this exquisite control to fine-tune exactly how much protein gets made from any given gene, adjusting the brightness for any situation. And the journey these tiny molecules take is just incredible. What you're seeing here is the whole complex assembly line that transforms them from just a piece of genetic code inside the cell's nucleus into these powerful regulators out in the cell's main workspace, the cytoplasm. So, how do you make one of these genes silencers? It's actually a pretty cool multi-step process. First, a long precursor RNA is made in the nucleus. Then it gets trimmed down and shipped out. Once it's in the cytoplasm, an enzyme with the perfect name, dicer, cuts it into its final tiny form. This mature mRNA then gets loaded into a special protein complex. And now it's armed and ready. This complex patrols the cell, finds its target messenger RNA, and latches on, either blocking it from doing its job or just marking it for complete destruction. It's a remarkably precise system of control. Okay, so if microRNAs are the dimmer switches, then our next characters, the long non-coding RNAs, well, they're more like the grand conductors of the entire genomic orchestra. These molecules are much, much larger and way more complex. They are true multitaskers. They can physically interact with DNA, with proteins, and even with other RNA molecules to control genes on a massive scale. And they do this in three main ways: by acting as guides, scaffolds, or decoys. First up, the guide function. Imagine you have a team of construction workers. Those are the proteins, but they have no idea where to build. The long non-coding RNA acts like a cellular GPS. It binds to that protein complex and physically drags it to the exact right spot on the DNA to either turn a gene on or shut it right down. Then there's the scaffold. Sometimes a bunch of different proteins all need to come together to get a big job done. A long non-coding RNA can act as a physical platform, kind of like a workbench, that brings all the necessary players together in one place so they can work as a single coordinated machine. And finally, the decoy. Now, this is a really clever bit of indirect control. A long non-coding RNA can basically act like a molecular sponge, soaking up other regulatory molecules like those microRNAs we just talked about. By trapping them, it stops them from silencing their usual targets, which has the effect of turning those target genes up. And this isn't just a theory. This has huge real-world consequences. Here's a powerful example of that guide function in action. A long non-coding RNA called hot air is often way overactive in cancer. It acts as a guide. It grabs a specific protein complex and drags it to genes that are supposed to prevent cancer from spreading. By bringing the complex there, hot air shuts down those protective genes, which allows the cancer to become much, much more aggressive. So understanding this hidden world of RNA isn't just some academic exercise. It's opening up entirely new frontiers in medicine, completely changing how we diagnose and even treat disease. Because these non-coding RNAs are so central to how a cell works, their levels change dramatically when you have a disease. This makes them incredible biomarkers. I mean, imagine getting a simple blood test that can detect cancer super early, predict how aggressive it's going to be, or even tell your doctor in real time if your treatment is actually working. That's the power we're unlocking with these RNA signatures. And we're not just diagnosing with RNA, we're using it to build entirely new kinds of drugs. So if a disease is caused by too little of a helpful microRNA, we can actually add more back in with something called a synthetic miRNA mimic. And if a disease is being driven by way too much of a harmful one, we can shut it down with a blocker called an antigomer. We're moving past treating symptoms and starting to actually rewrite the faulty software in our cells. So what this all really means is that we're moving way beyond the idea of DNA as just some static blueprint. We're now starting to see that our cells are actually run by this dynamic, complex, and incredibly powerful RNA-based operating system. This is the system that reads the blueprint and truly brings life to life. For years, we really thought we had read the book of life, but it turns out we may have only read the main text. We're just now starting to decipher all the footnotes, the annotations, and the editor's notes that are all written in RNA, and they might just change the meaning of the entire story.